Welcome to our program, Replacing Live Animal Testing with 3D Printing Technology. Over the past several decades, countless technological innovations such as 3D printing have been having a major impact on the world. 3D printing is an emerging revolutionary technology that could completely change the current approach to design and manufacturing with profound geopolitical, economic, social, demographic, environmental, and security implications. 3D printing technology is not only altering human life, but is also significantly impacting the lives of animals. It's exciting when physicians and scientists discover new ways to cure diseases and thus improve human health. Unfortunately, however, before reaching the final stage of development, numerous tests are conducted on animals that cause them great pain, suffering, and even death. Understanding this situation, a number of compassionate scientists are now developing ways to use 3D technology in place of animal testing. On today's program, we happily share some of these great technological advances with our noble viewers. We'll start off with a group of English scientists who have figured out a way to 3D print cells grown in a laboratory to create living structures. This approach could revolutionize regenerative medicine, enabling the production of complex tissues and cartilage that could potentially support, repair, or augment diseased and damaged areas of the body. Researchers at the University of Oxford and the Centre for Molecular Medicine at Bristol in the UK were able to demonstrate how human and animal cells can be printed into high-resolution tissue constructs. We were aiming to fabricate three-dimensional living tissues that could display the basic behaviors and physiology found in natural organisms. To date, there are limited examples of printed tissues, which have the complex cellular architecture of native tissues. Alexander Graham, lead author and 3D bioprinting scientist at Oxford Synthetic Biology, said in a press release, "Hence, we focused on designing a high-resolution cell printing platform from relatively inexpensive components that could be used to reproducibly produce artificial tissues with appropriate complexity from a range of cells, including stem cells." The researchers hope that with further development, the materials will have a great impact on healthcare worldwide. In the United States, Harvard University researchers have also developed 3D printed organs as an alternative to animal experimentation, having successfully made the first 3D printed organs on chips. This new technology should help to reduce the rate of drug testing on animals by pharmaceutical companies and academic labs, as the organs on chips provide a groundbreaking substitute for traditional ways of testing. Statistics from a study conducted by the website statisticbrain.com found that in the US, 19.5 million animals are killed each year for research. This is the first entirely 3D printed heart on a chip with integrated sensing. Organs on chips mimic the structure and function of native tissue. They are promising alternatives to traditional animal testing, but fabrication and data collection can be expensive and time-consuming. Now Harvard researchers have built the first entirely 3D printed heart on a chip with built-in sensors to track the beating tissue. The device is made using multi-material 3D printing in a single automated procedure. The process integrates 6 custom inks at micrometer resolution. Integrated sensors measure the contractile strength of the muscle, making it easier to study how tissue responds to drugs and toxins. 3D printed organs on chips with integrated instrumentation make fabrication faster and reliable data collection easier, paving new ways to study human health and disease in the laboratory. Our microfabrication approach opens new avenues for in vitro tissue engineering, toxicology, and drug screening research, says Dr. Keith Parker, senior co-author of the study, who is a WIS Core faculty member and TAR Family Professor of Bioengineering and Applied Physics at the Harvard John A. Paulson School of Engineering and Applied Sciences, or SEAS. Researchers are often left working in the dark when it comes to gradual changes that occur during cardiac tissue development and maturation because there has been a lack of easy, non-invasive ways to measure the tissue functional performance, says Dr. Johann Ulrich Lind, first author of the paper and postdoctoral fellow at SEAS. These integrated sensors allow researchers to continuously collect data while tissues mature and improve their contractility. Similarly, they will enable studies of gradual effects of chronic exposure to toxins. Supporting the use of technology to replace animal testing, Dr. Warren Casey, director of the U.S. National Toxicology Program's Interagency Center for the Evaluation of Alternative Toxicological Methods, which studies alternatives to animal use in chemical safety testing, says, in theory, non-animal tests could be much cheaper and much faster. Testing on these simplified, miniaturized versions of our physiology could deliver more human-relevant results than animal experiments. Significantly, the test could also replace the use of whole animals in the exploratory stages of early research, when scientists don't necessarily need to test on whole systems. Organs on a chip, for the most part, address a single output or endpoint. Dr. Casey says, because all that may be required at this early stage is to test the behavior of one cell type in response to a drug or disease as a way to guide future research. Replacing this with a harm-free model is now a reality. We'll now take a moment to thank the scientists and researchers working on ways to replace animal testing for their efforts to care for both human and animal health and wellness. Please stay tuned to Supreme Master Television. Welcome back to our program, Replacing Live Animal Testing with 3D Printing Technology on Golden Age Technology. In the field of cancer research, the idea that scientists can disrupt cancer growth by changing the environment in which cancer cells divide is growing in popularity. The traditional way that researchers have tested this theory is to conduct experiments with animals. However, researchers at Michigan Technological University studying cell scaffolding aim to replace animal testing in cancer research with electrospun synthetics. Synthetic cell scaffolds simulate tumor cells and tissue, offering a more reliable way to experiment on new cancer drugs. A team led by Dr. Smita Rao, Biomedical Engineering Assistant Professor at Michigan Technological University in Michigan, USA, is seeking better ways to test cancer drugs before human clinical trials, to detect toxicities or other adverse effects, and to better understand cancer cell growth and migration. Her team, comprised of both graduate and undergraduate students, investigated producing simulated cancerous tumor tissue with fine nanoscale polymer fibers, starting with the extracellular matrix or scaffold, for later seeding with live cancer cells. In my lab, the focus has been on standardizing the process and using synthetic materials to keep the same chemical formulation of a scaffold, but change the physical structure of the fibers that are produced. Dr. Rao says, noting that changing the type of polymer or adding solvents to polymers introduces too many variables, which can affect the way cells grow on the scaffolds. Dr. Rao and her fellow researchers, therefore, can compare separate cell lines with different scaffold alignments by changing just one aspect of the experiment: voltage. By changing the voltage at which the polymer is spun, the researchers can alter the shape of the scaffolds, whether honeycomb, mesh, or aligned. Dr. Rao's team recently published an article in Royal Society of Chemistry Advances about manipulating electrical fields to achieve different scaffold patterns. Her team is working with the Baltimore, USA company Dipole Materials to explore scaling up the process. We can study why and how cancer cells metastasize. Dr. Rao says, we can understand in a true 3D system why pre-metastatic cells become metastatic, and provide tools to other researchers to study signaling pathways that change between pre-malignant and malignant cells. Continuing the trend, People for the Ethical Treatment of Animals, or PETA, one of the most well-known animal rights organizations in the world, helped save animals with a new 3D lung model. In an impressive scientific breakthrough, funding from the PETA International Science Consortium Limited has helped lead to the creation of a first-of-its-kind human cell-based 3D lung model that will be used to study the effects of chemicals and other substances on the deepest part of the human lungs. Comprised of human primary alveolar cells, fibroblasts, and endothelial cells, with or without macrophages. One side of the model can be exposed to airborne test material while the other receives nutrients from a liquid similar to blood. The model was responsive and durable. Repeated exposure over 3 weeks resulted in effects on barrier integrity and release of pro-inflammatory and fibrotic markers. Cultures containing macrophages demonstrated a delayed pro-inflammatory response compared to those without. As many animals are commonly used and killed in inhalation tests, this exciting development has the potential to save tens of thousands of lives. And because human physiology is very different from that of rats and mice, this new human-based model will better predict what happens when we breathe in chemicals. We're deeply grateful for the recent technologies involving 3D cell culturing and printed organs that are leading to new ways of refining, reducing, and ultimately eliminating animal testing altogether. We thank all the scientists and researchers who are working tirelessly to both improve human life and to protect the lives of our animal friends. May heaven bless your loving hearts forever.